Okay, so welcome back to this next video in which we are discussing Alzheimer's disease and Alzheimer's disease medication. Okay, right. So we've discussed that Alzheimer's disease is a neurodegenerative process, and we've just been discussing how the early symptoms of Alzheimer's disease can be accounted for by certain areas of the brain being particularly susceptible to this neurodegenerative process. Okay, so the first symptom of Alzheimer's disease is the loss of declarative memory, deficits in declarative memory, and we believe that this is because the hippocampus and the entorhinal cortex are particularly susceptible to neurodegeneration by Alzheimer's disease, okay? And they're going to degenerate first, which then leads to the deficits in declarative memory seen before anything else, okay? Then, going a little bit further on, the early cognitive decline is believed to be caused by the degeneration of the basal forebrain nuclei, okay, which are also particularly susceptible to neurodegeneration by Alzheimer's disease. Okay, so they degenerate, that leads to a reduction in cholinergic neurotransmission in the cerebral cortex, and that causes cognitive decline. Okay, right. Of course, in the later stages of the disease, when you've got full-blown dementia, that is no longer merely explained by the loss of cholinergic neurotransmission. That's because the cerebral cortex itself is now starting to die, basically, degenerating itself, okay? And that causes the full-blown late stages of the disease. Okay, right. So, what we now want to turn our attention onto is what is understood of the biochemical causes of the neurodegeneration. So remember, when we studied the histology of neural tissue affected by Alzheimer's disease, we saw these two cardinal features. We saw the amyloid plaques and we saw the neurofibrillary tangles. Okay, we now want to see what underlies the formation of the amyloid plaques and the neurofibrillary tangles. Okay, now the amyloid plaques are believed to occur first, and then they cause the neurofibrillary tangles, and then both of these two features together are then believed to kill the neurons. Now how? We don't know, I'm afraid. However, when we come on to discuss the Alzheimer's disease medication, we will discuss a drug known as memantine, okay? And by looking at the mechanism of memantine, that will offer clues as to how potentially the amyloid plaques and the neurofibrillary tangles are going to kill neurons. Okay, so we'll see that right at the end. Okay, right. Now then, we're going to start off then with what causes the formation of amyloid plaques. Now, before we do that, it's important at this point to now distinguish between two different forms of Alzheimer's disease. Okay, so there are two different forms known as sporadic Alzheimer's disease, which I'll just abbreviate to AD, and then there's also familial Alzheimer's disease. Okay, now what's the difference between these two? Sporadic Alzheimer's disease is when it occurs late in life. Okay, so this is also called late onset Alzheimer's disease. Now what does late onset mean? Well, generally it means 7th decade. Okay, so you're going to get Alzheimer's disease in your 7th decade of life, then that's called sporadic Alzheimer's disease, okay, or late onset Alzheimer's disease. This is the major form of Alzheimer's disease. Most people who get Alzheimer's disease get it in their 7th decade of life. Okay, familial Alzheimer's disease is much, much rarer than sporadic Alzheimer's disease. This occurs earlier than the 7th decade, basically, and is therefore called early onset Alzheimer's disease. Okay, and I want to stress once again how rare familial Alzheimer's disease is. It's unlikely that, you know, even if you think Alzheimer's disease runs in your family, it's unlikely that it's actually familial Alzheimer's disease. Unless it's occurring before the 7th decade of life, then it's not really considered familial Alzheimer's disease. Now, in familial and sporadic Alzheimer's disease, the same core histopathology occurs. You get the formation of the amyloid plaques and the neurofibrillary tangles, and these seem to lead to neurodegeneration. Okay, it's just when it occurs that's different between the two. Okay, now, in familial Alzheimer's disease, in people where you're getting Alzheimer's disease before the 7th decade, you can find mutations in single genes that are causing the formation of the amyloid plaques. Okay, that is not the case for late onset or sporadic Alzheimer's disease. Okay, however, the amyloid plaques are the same. They're made from the same thing between these two diseases. Okay, it's just we know exactly why they form in the case of familial Alzheimer's disease, whereas we don't know why they form in sporadic Alzheimer's disease. Okay, but we'll come to that when we come to it. Okay, so now let's talk about where the amyloid plaques actually come from. What are they made out of? Now, the starting point for this entire story then is with a protein known as amyloid precursor protein, okay, which is often abbreviated to APP for short. Okay, now, amyloid precursor protein is a single-pass transmembrane protein that is expressed in a huge number of different tissues, not just neural tissue, is an important point to emphasize. This is not just made in brain cells, it's made all over the body. Okay, now let me show it. Okay, so let's represent the cell membrane here. So these two parallel lines, these represent the inner leaflet and the outer leaflet of the cell membrane. So this is the extracellular fluid here, the ECF, and this is the cytoplasm here. Okay, now, amyloid precursor protein, or APP, is a single-pass transmembrane protein. Okay, so it's going to have a single membrane-spanning alpha helix. Now, it has its amino terminus extracellularly, and now I'm going to represent the polypeptide, this polymer of amino acids, as a line here. And then it's going to pass the membrane, and when it does pass the membrane, of course, you'll have a membrane-spanning alpha helix here. Okay, a spring-like structure. I'm just too lazy to actually show that. Okay, and then it will be in the cytoplasm down here with a C-terminus here. Okay, so this represents our amyloid precursor protein then. Now, amyloid precursor protein has a very special region, okay, within it, which I'm going to highlight in orange. Okay, and this is the dangerous region. This is the region that can be cleaved and out of the amyloid precursor protein and can aggregate into these amyloid plaques, which we'll discuss later. Okay, so this bit's known as the Abt region, which stands for amyloid beta region. So this stands for amyloid beta region. Okay, right. Okay, so what we now want to discuss is how amyloid precursor protein is cleaved. Okay, so this protein is going to be cleaved up into different bits, basically, by a bunch of different enzymes. Now, there is a safe way that it can be cleaved up, and then there is a dangerous way that it can be cleaved up. So we're going to firstly talk about the safe pathway by which you can cleave amyloid precursor protein. And if you cleave it in this way, then it doesn't produce anything that's going to aggregate into amyloid plaques. We will then move on to the dangerous pathway for cleaving amyloid precursor protein. Okay, so then, we'll start with the safe pathway. Now, the safe pathway is going to firstly involve enzymes that are now called alpha secretases. Okay, now there is not just one alpha secretase. Okay, alpha secretases are defined because of where they cut amyloid precursor protein. Now, many different enzymes have been have been found to cut amyloid precursor protein at this site, okay, and therefore are counted as alpha secretases. So where are alpha secretases going to cut? Well, they're going to cut right in the middle of this amyloid beta region, okay, still on the extracellular side. Now, you will notice, actually, I should have stressed this more when I actually drew the amyloid beta region, that part of the amyloid beta region is within the membrane spanning alpha helix, okay. Alpha secretase is going to cut on the extracellular aspect, so it's going to cut before you actually go into the membrane spanning alpha helix portion. Okay, right, so there's a specific site where alpha secretase alpha secretases cut amyloid precursor protein. Okay, and in order to be considered an alpha secretase, all you have to do is cut amyloid precursor protein at this specific point. Okay, so there are loads of enzymes that are defined to be alpha secretases by the definition of what it is that you have to obey to be an alpha secretase. Okay, however, there is a main alpha secretase enzyme. There is one that we believe that is most involved in doing this. Okay, now this enzyme that we believe is most involved in alpha secretase activity is known as ADAM10. Okay, and in fact all the other alpha secretases that we've found are also members of this ADAM family of enzymes. Now what does the ADAM stand for? Okay, well it stands for A, disintegrin and metalloproteinase. Okay, so the A is for just A, and then the D is for disintegrin, the second A is for and then the M is for metalloproteinase. Okay, and it's a massive great family of enzymes that are involved in cutting up portions of the extracellular matrix and are therefore involved in the remodeling of the extracellular matrix. Okay, so this specific member of this family of A disintegrin and metalloproteinases, ADAM10, is believed to be the cardinal alpha secretase, basically. But other members of this same family, other ADAM enzymes, are also alpha secretases. They also show alpha secretase activity. It's just this one seems to be the most important. Okay, so ADAM10 then. Firstly, we don't have a very clear picture of what the structure of ADAM10 is, which is a shame. What we do know is that it is an integral membrane protein, but because we don't really have that clearer picture of what its structure actually is, I'm just going to draw it like so. Okay, here, this portion is the portion attaching it into the membrane, and then here is its actual active site portion, which is on the extracellular aspect. Okay, like so. And it doesn't just cleave amyloid precursor protein, it will cleave all sorts of other things, okay, other components of the extracellular matrix. Okay, so this here, this represents ADAM10 here in green. Okay, or another alpha secretase, another one of these ADAM family proteins. Okay, so what it's going to do then is it's going to cut amyloid precursor protein, which is in the cell membrane of our cell here. And now we're imagining that this is a neuron, but this will also be happening elsewhere in the body as well. Okay, so it's going to cut here, and then what's going to happen is you're going to get two fragments produced then. One that's going to be secreted extracellularly. Okay, so here's the amino terminus, then you've got that top portion of the Abt region here. Okay, now what's this fragment known as? It's known as the amyloid precursor protein, and then you put S for secreted, and then you put alpha because it's being cleaved by the alpha secretase enzyme, or it's being cleaved at the alpha secretase cleavage site here. Okay, right, so this stands for amyloid precursor protein, which I won't write out in full, and then secreted fragment alpha, okay, because it's now being secreted out into the extracellular fluid. Okay, right, so that's this first fragment that's produced by amyloid precursor protein when it's being cleaved in this safe pathway. Now, the other fragment is still in the membrane, okay, and is now going to be acted upon by another enzyme, okay, known now as gamma secretase. Okay, so now what we need to discuss is what is gamma secretase. Now, gamma secretase, I'm afraid, is even more complicated than alpha secretase. Gamma secretase, unfortunately, is not just one protein. It's a multi-protein complex consisting of 4 separate proteins stuck together. And I'm going to bore you now with the structures of all 4 of them. Okay, right, because they do become important, okay? If you read the literature on Alzheimer's disease, you will hear all the time about mutations in presenilins, okay? And if you don't know the components of gamma secretase, you won't understand what a presenilin is, okay? So we're going to discuss the components of gamma secretase. Okay, so there are 4 components then of gamma secretase, and they're all integral membrane proteins. Okay, so we will start off with the one that actually catalyzes the cleavage. Okay, all the other three are just sort of stuck on the edge, and they sort of help out, basically. But the one that actually catalyzes the cleavage is the presenilin protein. Okay, spelt like so, presenilin. Okay, now there are two different presenilins in humans. There's a presenilin 1 and there's a presenilin 2, and they can both function in the same way. Okay, so there's presenilin 1 and presenilin 2, and they're often abbreviated to PS for short. So you'd have PS1 is presenilin 1, PS2 is presenilin 2. Okay, so let's show the membrane spanning topology then of a presenilin protein. Okay, so they have 8 membrane spanning alpha helices. Now 6 of them are all clustered together. Okay, so these are the first 6 all clustered together, and then the final 2, the 7th and the 8th one, are then on their own with a large intracellular loop between 6th and 7th. Okay, so this is the membrane spanning topology then of a presenilin protein. So we'll have presenilin here shown in green. And this is the one that's actually going to catalyze the cleavage of the fragment that is left over of the amyloid precursor protein after the alpha secretase ADAM10 has done its job. Okay, right. So this is our presenilin, and it's either presenilin 1 or it's presenilin 2. Okay, then the next component of gamma secretase is a single membrane spanning protein here. Okay, and this is known as nicastrin. Okay, so this protein is called nicastrin. So we'll colour in nicastrin in orange here. Okay, and its function is accessory to presenilin. Okay, so it sticks on the side and performs some sort of function that helps out. I think nicastrin is believed maybe to have a role in recruiting the substrate, in grabbing the substrate and giving it to presenilin effectively. Okay, but I think this sort of stuff is still controversial. Okay, next up, the next protein is known as APH1. Okay, and it's got 7 membrane spanning alpha helices, but before you think, oh, it's a G protein coupled receptor, no. It's got its carboxylic acid terminus extracellularly and its amino terminus intracellularly. That's the wrong way round for being a G protein coupled receptor. Okay, so it's not a G protein coupled receptor. Okay, this is APH1 here. Okay, and this stands for anterior pharynx defective 1. Okay, so the A is for anterior. Okay, the PH is for pharynx. Okay, and then it's defective, and then you'll have a dash between pharynx and defective so that it's effectively one word, and that explains why the defective doesn't get a symbol in this abbreviation. Okay, so this is our anterior pharynx defective protein here in blue, and again its function is accessory to the presenilin. Okay, so here it is in blue. And then the final protein in this gamma secretase complex is a protein known as PEN2. Now PEN2 has 2 membrane spanning alpha helices, like so, with the amino and the carboxy termini both on the extracellular side of the membrane. We'll colour it in pink here. Okay, so this protein is PEN2. And PEN2 stands for presenilin, that's the P, and then it's presenilin enhancer 2. So the EN is for enhancer. Okay, so presenilin enhancer 2. Right, and I'll underline its name in pink as well. So these are the 4 proteins which make up a gamma secretase enzyme. Okay, right. So what's going to happen is this great multi-protein complex is now going to cleave what we've got left. Okay, and I'll draw what we've got left down here then. Okay, so here is our membrane again. Okay, and here is our amyloid precursor protein, or what's left of the amyloid precursor protein after the alpha secretase enzyme has worked. You've got the bottom portion of the amyloid beta region, and then you've got this cytoplasmic portion here. Okay, now, gamma secretase actually cuts within the membrane spanning alpha helix region of the amyloid precursor protein. Now the other thing is that it has 2 different sites at which it can cut. Okay, one site which we'll have here, and then the other site here. Okay, so one of the sites that it can cleave is right at the end of the amyloid beta region, and then the other site is 2 amino acids up from that. Okay, so basically, if we zoom in on this, what you have effectively got is these 2 cleavage sites, and in between them there are two amino acids. So here is one amino acid and here is the second one. Okay, and this is still all part of this amyloid beta region. Okay, so you've got these two sites, and depending on which one you cut at, you're going to either add two amino acids onto this fragment, or you're going to add two amino acids onto the other fragment. Okay, so there are two slightly different sites at which the gamma secretase can cut. Okay, now, when it cuts then, it will cut at one of these, and then it's going to produce two fragments. Okay, so let's draw the two fragments. One is going to be a secretive fragment, so the remaining orange portion, okay, here, like so, fragment is going to get secreted, and the other fragment is going to drop into the cytoplasm here. So here's the amino terminus of this fragment, here's the amino terminus of this fragment, and the carboxy terminus of this fragment. Now, depending upon your site of cleavage, okay, that will determine how long this fragment is. If you choose the first site of cleavage, okay, then this one's going to be slightly shorter, and this fragment here is going to have two additional amino acids on it, the two amino acids from this little bit here. Okay, so the two final amino acids of the amyloid beta region, basically. Okay, then if you instead decide to cut right at the end here, that will mean that this little fragment becomes longer, and then finally this fragment will be slightly shorter. It will not have these two amino acids here. Okay, however, no matter whether they, the naming of these two fragments isn't determined by whether or not they have the two additional amino acids here. So we name these fragments the same despite where the two amino acids have ended up, basically. Okay, so this fragment is always called the P3 peptide, whether it has got these two amino acids or not, basically. Okay, so this is called the P3 peptide, and then this portion that's dropped into the cytoplasm, this is known as the AICD. Okay, so what does that stand for? That stands for the amyloid precursor protein, that's the A, and then the ICD stands for the intra, that's the I, cellular, that's the C, and then domain, that's the D. So the amyloid precursor protein intracellular domain. So here is the amyloid precursor protein intracellular domain, the AICD, which is dropping into the cytoplasm. Okay, so that then is the safe pathway by which you can cleave up amyloid precursor protein. Okay, none of these things are believed to be harmful. Okay, amyloid precursor protein secreted fragment alpha, which was the first bit produced, that's not believed to be harmful. In fact, that's believed to be very beneficial. Okay, P3 peptide, that's not believed really to be harmful, and neither is the amyloid precursor protein intracellular domain. Okay, now, there is another way of cleaving up amyloid precursor protein which is harmful. Okay, so the bad way of cleaving up amyloid precursor protein. Now, this is the important bit with regards to the sporadic and the familial Alzheimer's disease. What is going to happen is, for some reason in neurons, you are going to stop cleaving up amyloid precursor protein by the safe way, and instead you are going to start cleaving it up by the unsafe way, okay, which is going to result in the formation of amyloid plaques. Okay, now, in the sporadic cases of Alzheimer's disease, we do not know what causes you to change from from cleaving it in this way to cleaving it in this unsafe way. Whereas in the case of familial Alzheimer's disease, we do know what causes you to start using the unsafe way rather than the safe way. Basically, in many cases of familial Alzheimer's disease, mutations in amyloid precursor protein have been found, which result in the amyloid precursor protein being more likely to be cleaved by the unsafe way rather than the safe way. Okay, so that's where sporadic and familial Alzheimer's disease differ. We know why the unsafe pathway is used in familial Alzheimer's disease, okay, and therefore we can explain why the amyloid plaques form. Whereas in the sporadic cases of Alzheimer's disease, we don't know what fundamentally causes you to stop using this safe pathway and start using this dangerous pathway. Okay, right, so let's now discuss the dangerous pathway and what it's going to produce. So once again, let's draw out our amyloid precursor protein again. Okay, so here is our amyloid precursor protein. Here's the amino terminus, here's the carboxylic acid terminus, and then you've got the amyloid beta region here in orange. Okay, right, so now instead of the alpha secretase enzyme cleaving the amyloid precursor protein, instead what's going to happen is an enzyme known as beta secretase is going to cut the amyloid precursor protein. Okay, now, once again, just like alpha secretase was defined by cutting at a specific site rather than pointing at a protein and saying that's what we're going to call alpha secretase, beta secretase is again defined just as an enzyme that cuts amyloid precursor protein at a certain site. So again, there could be loads of beta secretases. However, we think that there is only one beta secretase. Okay, and this specific protein that is the beta secretase in humans is known as base 1. Okay, now what does that stand for? That stands for beta site amyloid precursor protein cleaving enzyme 1. Okay, so this here is for beta site. Okay, so that's supposed to be the Greek letter beta. So beta site, and then the A is for amyloid precursor protein, APP. Then the C is for cleaving, and the E is for enzyme. And then it's the beta site amyloid precursor protein cleaving enzyme 1. Okay, right, so I'll underline this in pink here. Right, so where is the beta site for cleavage? Where do beta secretase enzymes cut the amyloid precursor protein? Well, it's right at the beginning of the amyloid beta region here. So this is our amyloid beta region here. So beta secretases are defined as enzymes that cut right at the beginning of the amyloid beta region of the amyloid precursor protein. Okay, and the beta secretase that we have found in humans is this base 1 enzyme. Okay, so let me show you the structure then of base 1. So base 1, its structure is known slightly better than Adam 10. Okay, so it's a single membrane spanning protein, and it has its enzymatic portion extracellularly, and then it has a single membrane spanning alpha helix attaching it to the cell membrane, and then the carboxylic acid terminus is intracellularly. Okay, whoops, I was about to colour it in blue, but I think I should probably colour it in pink so it's colour-coded. Okay, so for some reason you're going to switch from processing amyloid precursor protein in the safe way, and now you're going to start processing it in the unsafe way. And the first cleavage that's made in the unsafe way is that this beta secretase enzyme, base 1, is going to cut the amyloid precursor protein here. Okay, now that's going to create a soluble fragment again. Okay, so this portion that's being produced here, which now has no portion of the amyloid beta region within it. Okay, this is known as the amyloid precursor protein secreted fragment beta now. Okay, so contrast that to the amyloid precursor protein secreted alpha fragment. The alpha fragment had a lump of the amyloid beta region attached to it. Okay, right. Then the portion that we've got left over now is going to be cut by the gamma secretase, okay? And the gamma secretase is going to still cut at the same site. So remember, there were two sites where the gamma secretase could cut, which differed by 2 amino acids, okay? So the gamma secretase is going to cut at this first site, creating a slightly shorter amyloid beta fragment now, okay? So this... Ooh, and I'm showing it going off in the wrong place. How am I going to do this? So this should be going extracellularly, but I haven't got space to show... Well, I'll do it up here. Okay, I don't want to imply that it goes into the cell. It's going extracellularly. Okay, so if the gamma secretase cuts at the first site, you'll get this slightly shorter protein, which is now called amyloid beta, okay, 40. Okay, so it's now an amyloid beta peptide, and it's called 40 because it contains 40 amino acids. So from this starting portion all the way down into the middle of the membrane spanning alpha helix portion here, you have 40 amino acids, and that's the amyloid beta 40. Now, if gamma secretase cuts at the slightly further along position by 2 amino acids, you'll get a slightly longer amyloid beta peptide here. Okay, and that's known as amyloid beta 42 peptide. Okay, right, and then the other thing that you'll produce once again is a amyloid precursor protein intracellular domain here. the AICD, which is identical to what it was in the safe pathway. Okay, right, so these are the dangerous things that you have now created in this pathway. Why is this pathway dangerous? Because it creates these amyloid beta peptides. Okay, so these are known as the amyloid beta peptides, and these are what are going to cause the formation of the amyloid plaques. Okay, right, so there is a fancy word for these amyloid beta peptides, okay, that describes them. They are amyloidogenic. They have a tendency to aggregate and form amyloid plaques. Now, particularly amyloid beta 42 is terrible for doing this. It is incredibly amyloidogenic. Okay, amyloid beta 40 is not quite as bad. It's not good, but it's still amyloidogenic, but it's nowhere near as amyloidogenic as amyloid beta 42. Okay, so gamma secretase cutting at this lower site is more dangerous than it cutting at the top site, basically. Okay, now this brings us to a few more mutations that are seen in familial Alzheimer's disease. So, other mutations that are seen in familial Alzheimer's disease. So, some people will have mutations in amyloid precursor protein that just make the amyloid precursor protein more likely to be processed by this dangerous pathway. Some people will then have mutations in amyloid precursor protein that actually mean that when gamma secretase cuts it, it is more likely to cut at the lower site, and therefore if you are processing amyloid precursor protein by this dangerous pathway, you're more likely to be producing amyloid beta 42 than amyloid beta 40, and if you overproduce amyloid beta 42, that's worse than producing more amyloid beta 40, so you get amyloid plaques forming quicker, basically. Okay, explaining the earlier onset of the disease. In addition, other mutations that have been found in familial Alzheimer's disease are mutations in the presenilin proteins. Okay, so remember the presenilin proteins were this component of the gamma secretase enzyme. Okay, and they were the component that actually does the cutting. Now, some people have been found to have mutations in the presenilin proteins that mean that the presenilin proteins prefer to cut at the lower site than they do at the higher site, and therefore they prefer to produce amyloid beta 42 compared to amyloid beta 40. So those are other mutations that then lead to early onset or familial Alzheimer's disease. Okay, right. Now, let's then discuss how these amyloid beta peptides are going to aggregate together to form an amyloid plaque then. Okay, so basically what happens is these amyloid beta peptides fold round to form something known as a beta peptide. Okay, so we'll have the carboxy terminus here and the amino terminus here. Okay, so this is what's known as a beta hairpin. Okay, so this is called a beta hairpin, and it's held together by beta interactions between these two parallel strands. So let me just remind you what beta interactions between two parallel strands are. So this is a secondary protein structure, basically. It's going to be held together by interactions between the peptide links between amino acids within the polypeptide. So let me just draw a small portion of a polypeptide. Okay, so let's say here, this is the amino group of an amino acid here. Then we've got the alpha carbon here, okay, with a hydrogen coming off it. We'll put an arbitrary R group here. Then we'll have a carboxylic acid group here, okay, like so. And then you'll go on and on and on. So I've just drawn one of those amino acids here. Okay, right. Now, when you twist back around then, when the polypeptide twists back around, the way this is going to now be organised is that in this parallel strand, you'll have the amino group facing in like so. Okay, then you'll have the alpha carbon here, maybe with its hydrogen coming off, its R group maybe coming off down here, and then the carboxylic acid group coming off down here. Okay, now the idea is that the amino group on this parallel strand is going to interact with the carboxylic acid group on this parallel strand here, and it's going to be doing the same for other amino acids further along here as well. Okay, so how is the amino group of this amino acid here going to interact with the carboxylic acid group here? Well, it's by hydrogen bonds. Okay, so what are the two things that you need to form a hydrogen bond? Well, firstly, you need a hydrogen bond donor. Okay, so this hydrogen coming off the nitrogen is going to be a hydrogen bond donor. So let me explain what that means. So basically, in order to be a hydrogen bond donor, you need a hydrogen that is attached to an atom which is far more electronegative. Now, what does electronegative mean? Well, let me just remind you of what electronegative means. So, in a covalent bond, such as this covalent bond that we have between the nitrogen and the hydrogen, you have two electrons. Okay, one from the nitrogen and one from the hydrogen. Okay, now the question is, do these two electrons sit right in between the nitrogen atom and the hydrogen atom? Okay, and the answer is no. Okay, they are going to sit slanted towards the nitrogen atom. Okay, and the reason is that the nitrogen atom is going to attract these electrons more than the hydrogen atom. Okay, now, why does the nitrogen atom attract the electrons more than the hydrogen atom? Well, what does the nitrogen atom have that will be attracting electrons? Well, in the nucleus of atoms, you have neutrons and protons. Neutrons are neutrally charged, but protons are positively charged. Okay? Protons, therefore, are going to be exerting a pull on the electrons, which are negatively charged. So the protons of the nitrogen atom are going to be pulling the electrons in this bond towards them. Now you might say, okay, so it's just a case of which one has more protons, and then of course the nitrogen's bigger than the hydrogen, so it has more protons, so now it's obvious that the electrons are going to be pulled towards the nitrogen. But don't be so hasty, it's a little bit more complicated than that, because you also have to consider the inner electrons, the electrons that are shielding the positive charge of the nucleus from these electrons that are in this bond here. Okay, because those electrons will be repelling these electrons here. So it's much more complicated working out whether an atom, well, how strongly an atom is going to pull electrons towards it. Okay, but we have a big word that describes how strongly an atom pulls electrons in bonds towards it, and this is electronegativity. Okay, and it's a well-known fact in chemistry that nitrogen has a very high electronegativity. Its ability to pull electrons towards it is very, very good. Okay, its electronegativity is very strong, whereas hydrogen has a sort of much lower electronegativity, and therefore nitrogen's pull on these electrons is going to be greater than hydrogen's. So these electrons are going to sit slanted towards the nitrogen. Okay, right. So what's the result of that? Well, it means that the nitrogen develops a partial negative charge and the hydrogen develops a partial positive charge. Okay, so that is the feature of a hydrogen bond donor. You need a hydrogen atom attached to an atom which has a much higher electronegativity than hydrogen, and therefore produces a partial positive charge on this hydrogen here. Okay, and that's what you need to be a hydrogen bond donor, the hydrogen with a partial positive charge here. Now, you also need a hydrogen bond acceptor. Okay, and that's what the oxygen over here is going to be. So what are the characteristics then of a hydrogen bond acceptor? Well, in order to be a hydrogen bond acceptor, you need to be an atom which has a partial negative charge, firstly. So you need to have an atom that is far more electronegative than another atom, okay, and it needs to be bound to that other atom. So oxygen has a far greater electronegativity than carbon, so it's going to pull the electrons in these two bonds here towards it, which is going to give it a partial negative charge, and the carbon will therefore gain a partial positive charge. Okay. However, partial negative charge is not the only thing that you need to form a hydrogen bond. You also need at least one lone pair of electrons. Now this oxygen actually is going to have two lone pairs of electrons, but you don't need two lone pairs of electrons to be a hydrogen bond acceptor. You only need one, so I'm only going to show one. Okay, so these are the two features then of a hydrogen bond acceptor. You need a lone pair of electrons and the partial negative charge. Okay, now the idea is that the partial positive hydrogen is going to form a very nice, strong electrostatic interaction with this partial negatively charged area, which has a lone pair of electrons, which is a center of negative charge, because the electrons both have negative charges. Okay, and that's going to form quite a strong form of intermolecular bonding, and that bond that you've got there is known as a hydrogen bond. Okay, and they are reasonably strong. Okay, so you're going to be forming these all the way up and down these two parallel strands that you've got here. Okay, and that's what's going to be holding this structure in place. So that's what's known as a beta hairpin. So the amyloid beta peptides, amyloid beta 40 and amyloid beta 42, are going to be forming these beta hairpin structures. Now, beta hairpins can then aggregate into fibrils. So remember what a fibril is. A fibril is a little fiber. Okay, so how can the beta hairpins aggregate into fibrils? Okay, well what can happen is they can end up stacking on top of one another. Okay, so if I draw one beta hairpin here, and now I'm going to drop showing the C and N terminals because it will just get messy if I keep those on. Okay, here is our beta hairpin. What you're going to do is stack beta hairpins on top of one another. So here is another one stacked on top of this one, another one, and you're just going to continue on going, stacking more and more beta hairpins on top of each other, and this will make you a long fibril, basically, of beta hairpins all stacked on top of one another. Now, why can the beta hairpins stack on top of each other like this? What's holding them together? Why doesn't the fibril just fall apart? Well, basically, these beta interactions that we've talked about between these two strands are going to break, okay? And instead what you're going to get is beta interactions forming between the parallel strands on each side of the fibril. So all of these strands that I'm now showing on this side, in the green side here, those are now going to form beta interactions between one another, rather than you having beta interactions between the two strands within a single beta hairpin. Okay, so what's going to happen is they're going to break these interactions that are within a hairpin, and you're going to form new interactions between the hairpins. And of course you're going to do the same thing on the other side. Okay, so the side that I'm now colouring in pink here, that side is also going to get the formation of these beta interactions between strands that are in different hairpins. Okay, and then of course the hairpins are still going to be attached because they've actually got a bit of polypeptide linking them on either side. So you form this effective fibril that has these two beta sheets. Okay, so it's known as a double beta sheet, basically. It's two huge great beta sheets sitting on top of one another like so. Okay, and that will form a massive great fibril now. Okay, so that's how these amyloid beta peptides are going to aggregate into these fibrils. And then we know the story from here. Once you have a massive great fibril, which I'll now just represent as a line, but this is loads and loads of beta hairpins stacked on top of one another, what you can do is you can align loads of these fibrils together. Okay, and this is what's then known as an amyloid plaque. Okay, so that then is how you form these amyloid plaques in Alzheimer's disease brains. Okay, you process the amyloid precursor protein in this dangerous way. You produce these extracellular amyloid beta peptides, which aggregate into these fibrils, which then form amyloid plaques. Okay, so we'll call it there for this video, and in the next video what we'll move on to is discussing how the neurofibrillary tangles form, and we'll see how together the amyloid plaques and the neurofibrillary tangles are going to cause the death of neurons.